In the last class or in the last video, I have explained about the characteristics of genetic material, the types of nucleic acid and the compositions of the nucleic acid. Today, even we have already discussed even the structure of purines and pyrimidine, or the types of the purine and pyrimidine. Today we are going to discuss, in order to understand the structure of DNA, we have to understand how they are arranged and how they are placed, or in what form they are made up of. So last class I said that about the nucleotide and nucleoside. To understand the structure of DNA, you must understand nucleotide and nucleoside. So nucleotide is sugar, nitrogenous base, plus phosphate group, or you can say as phosphoric acid. Now your sugar is pentose sugar. Then nucleoside means if there is a sugar and the combinations of sugar and nitrogenous base is nucleoside. Now let's see in the form of chemical structure. I'll make a sugar, this is aged, OH, this is aged. So this is our pentose sugar. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. Now, I have said already in the last class that this in the carbon number 1, this is the position in carbon number 1, a sugar has to bind. In carbon number 1, this is sugar, sorry. So this is a nitrogenous base has to bind in the carbon number 1. So let me make one. It may be a purine or pyrimidine. Okay. So I'll make one pyrimidine. So this is pyrimidine. Just say this is thymine. Okay. Now. So how they form a bond is this OH and H. You have to eliminate water, H2O. So with the elimination of water, there will be a formation of bond between the sugar and the nitrogenous base. So let me form a bond. This H and OH is out. So this is H2O. So let me rearrange the structure. Just say that this is our formation of bond, nitrogen. So this carbon and this is sugar and this is nitrogenous base. So they form a bond. So this bond is known as N-glycosidic bond. Glycosidic bond. So these questions are usually asked in a competitive examinations for your NEET. And the next point you have to know is, so this sugar and nitrogenous base, we call this as nucleoside. Now the phosphate group will attach. Now this phosphate group attach at the fifth position. Let me make phosphate group or phosphoric acid. So the bond will form by elimination of water molecule. So this OH will be out and H. So this is, so OH plus H will be H2O. Okay. Now see, you can see that elimination of water, carbon number 5 of the pentose sugar and the phosphoric acid, they form a bond. This bond is known as phosphoester bond. Okay. Now I will consider this as one nucleotide. Okay. One nucleotide. One nucleotide means sugar. Sorry, this is a sugar, nitrogenous base, and phosphate. That will be considered as one nucleotide. Now let me make a chain extension. Okay. Now, as I said last class also, the phosphate group has to be attached at the 5 prime. This is the 5th position of the sugar always. Okay. Now, if I have to arrange the chain, if I have to elongate the chain or increase the number of nucleotides, then another phosphate group has to come. Let me make another phosphate group. OH, OH. Okay. Now, same thing. Here is a hydroxyl group, OH and H. So there will be elimination of water and the carbon number 3. Okay. So this is the carbon number 3. The carbon number 3 of the pentose sugar and the phosphate group. You can see another bond forming, and this bond will be considered as phosphoester bond. Now, once the phosphate group attached, what will come? Sugar, pentose sugar will come. So let me make another pentose sugar, H, and let me make one purines. Okay. Last time I have shown you the structure of purines. Okay. So this carbon number 1 of the pentose sugar and the pyrimidine. See, they will form a bond. Okay. And this is H and this is OH. Let me join what is here. This is carbon number 4. So this position will be carbon number 5. So same thing, they will take OH from this carbon number 5 and the H. Now they will form a bond. Okay. So this bond is said to be N-glycosidic bond or N-glycosidic linkage. And this bond will be considered as phosphoester bond. So together we call these two as phosphodiester bond. So if I consider this as one nucleotide, this will be another nucleotide. So it will keep on joining. Okay. So such a chain of nucleotides, we says that either DNA or RNA are made up of polymers of nucleotide. So you can see that this is 5 prime and this is, here is a third. So you will always see in the 5 prime as a phosphate group, and at the 3 prime you will see hydroxyl group. Okay. It will be same for this chemical composition will be same for both DNA and RNA. As I said last class also, that the only difference will be in the pentose sugar. If it is for DNA, the structure will be like this, okay, H. But if it is for RNA, then additional hydroxyl group will be present. Now, let me explain the difference between DNA and RNA. Okay. as I said that both having the same composition, the only difference is the pentose sugar. So this is the reason. In the first point is hydroxyl group. In DNA, you find only one hydroxyl group at the 3' position. But in RNA, you find additional hydroxyl group. So the more the number of hydroxyl group, the molecules become reactive. Okay. So one point is clear that the RNA is more reactive as compared to DNA based on the number of hydroxyl group. So DNA always, if we compare DNA and RNA, both undergo mutation, but here it will be slow and the RNA will be fast. Okay. As I said that it is slow. Why is it slow? Because slow mutations are required, okay, necessary for the process of evolution. Okay. Second point is stability. Now which one is more stable? Okay. Now in DNA, as I said that there is a thymine. Okay. You know that there is a thymine in DNA. So presence of this thymine, okay, it provides additional stability to the structure of DNA. But in RNA, you know that there is a uracil. Now, uracil are unstable structure, okay, unstable structure. Why unstable? Because uracil can convert to cytosine by the process of deamination. I'm repeating again. Why uracil is unstable structure? Because they can convert it to by the process of deamination, they can convert to cytosine. Okay. So what is deamination? Removal of NH2. Amino group get removed. Okay. So if uracil keep on changing its structure, it means that they are unstable, okay, and they will be more reactive. Okay. So presence of the thymine confers the additional stability to the structure of DNA. Presence of uracil in RNA provide unstable structure. Point number 3, that is the expression of information. This is very important. Expression of information. Now, what does it mean? Okay, you know that DNA, they store the genetic information. Whatever the parents transmit or whatever the parents have the character that has to be transferred to the next generation. So, the character that has been transferred were stored in DNA. Okay, they are the storage of the genetic information. Okay. Whereas the RNA, they are responsible for the transmission. Transmission means whatever the information that is stored in the DNA, that has to be transmitted. They have to be transferred. Okay. So RNA is responsible for that. So, but in the case of like expression for DNA, they have to convert from DNA to DNA, and they have to convert to mRNA, and they have to convert to protein. Okay. Whereas in case of RNA, okay, in case of RNA, RNA is converted to DNA, okay, then DNA to mRNA, and then mRNA to protein. So this is the difference. Okay. So you can see that DNA converted to DNA, then after DNA they become RNA, then protein. Whereas in case of RNA, RNA will be converting to DNA, then this DNA will convert to mRNA, and then mRNA get converted to protein. This is called reverse transcription. reverse transcription. You can see this example in the case of viruses, okay, where the RNA as a genetic material, they convert to DNA, then only they form mRNA, then protein, okay. So these are the three important differences between DNA and RNA. The structure of DNA. As I said that Friedrich Mesher was the scientist who had first seen the DNA in a past cells. But the structure of DNA was explained by Watson and Crick, okay, who got a Nobel Prize. Now, before that, there was two important scientists. Two scientists were responsible. I will give a credit to them first because they are the ones who have produced this structure, and they have given this data. After collecting the data, they have given to the Watson and Crick. Okay. So it was Friedrich Mesher who had given the idea about observe. Okay, he had observed the structure of DNA. Okay, and he named as nuclein, right? But he had observed, but he was not able to explain the structure of the DNA. So there was a scientist called as Maurice Wilken and Rosalind Franklin. Okay, so they have done the experiment and they have provided the data to Watson and Crick. X-ray diffraction of DNA. So through the X-ray diffraction, they have provided the data to Watson and Crick. Okay, Watson and Crick. Now, with this data given by the Maurice and Franklin, a double helical structure of DNA was given by these two scientists. So they have explained and they have given the postulates. Okay, double helical structure of DNA. Okay, this you have been studying from class 10. Okay, now before understanding the postulates of Watson and Crick, let's try to understand the three basic rules. Okay, that is Chargaff rule, base pairing rule, and the hydrogen bond. Now, what did he say? He said that adenine, that is your purine. Adenine is equal to thymine. So you know that adenine is purine and this thymine is pyrimidine. Okay, I'll represent as py. And he says that guanine is equal to cytosine. And you know that this is purine, and this is pyrimidine. So he said that adenine is equal to thymine, guanine is equal to cytosine. He also said in a second point that A by T is equal to 1, okay, and G by C is equal to 1. It means that whatever the amount of adenine, there must be an equal amount of thymine. Whatever the amount of guanine, there must be equal amount of cytosine. So in his statement he said that, okay, from that data, the Watson and Crick had explained the structure of DNA. Now let me give one example. Okay, if I have a 20% of adenine, what will be the percentage of just say guanine. Okay. So if I have 20% adenine, then there must be 20% of thymine. So 20 plus 20 is 40. So if there is a guanine, there must be cytosine. Okay. So remaining 40 means 60. So 30, 30. So that will give 60. So the percentage of guanine here will be 30%. Okay. So these small questions are asked in the competitive examinations. Okay, based on the Sargaf rule. The second important point is base pairing rule. Now, what is base pairing rule? It says that if adenine is there, there must be thymine. No other base can join, okay, can bind. If adenine, there must be thymine, and if there is a guanine, there must be cytosine. So likewise, if there is a thymine, there must be adenine, and if there is a cytosine, there must be guanine. Okay, so this is said to be base pairing rule, or we say complementary to each other. For example, if one strand 5' to 3' and 3' to 5', if one strand have adenine, the next strand, okay, will be thymine. Fine. So I've already discussed this, the structure of your DNA. It means that the nucleotides have been explained. Okay. So this site is called what? This side is sugar, pentose sugar, plus phosphate. Okay, so this is said to be your backbone of the DNA. Okay, so when I say backbone, it means that pentose, sugar and phosphate considered as the backbone of the DNA. Okay, and the one which is inside, okay, inside, that will be nitrogenous base. For example, here adenine, say guanine, thymine, okay, cytosine, C, T, G, A, A. Okay, see. So this, the one which is projecting inside, okay, that will be the nitrogenous base. So this is about the base pairing rule. And the third one is hydrogen bond. So hydrogen bond, in case of adenine and thymine, there must be only 2 hydrogen bonds. Always 2 hydrogen bonds. Whereas in guanine to cytosine, there must be 3 hydrogen bonds. Okay, so this 1, 2 and 3. Okay, this is considered as the hydrogen bond. Now, based on that, the Watson and Crick had explained and given the postulates. Let's see the postulates of the Watson and Crick model of DNA. The first point that they said was this is one strand and there will be another strand. It means that 2 polynucleotide chain. Poly means many, okay? So you can see that this is 2 polynucleotide chain or strand. Okay? So this is Watson and Crick model of DNA. I hope you have seen a staircase, okay, spiral staircase, okay. So if you go and play at this staircase, which is a spiral, you will understand how the structure of DNA are arranged, okay. So try to observe it, okay. So one will be the another one, okay? This is the another strand, another one, okay? The another strand is arranged in such a manner that it is a right-handed fashion, okay? It's a clockwise right-handed fashion. That is what the Watson and Crick had explained. Now, when I say two polynucleotide chain, this is if one runs from 5 prime, then it will be 3 prime end. So if it is the opposite strand should be 3 prime and the end will be 5 prime. Now it means that this is said to be anti-polarity. What is anti-polarity? If one strand runs from 5 prime to 3 prime direction, another strand will run from 3 prime to 5 prime direction. Okay, so that is the rule given by the Watson and Crick model of DNA. The third important point that you will see that they have already explained with the help of the Sargaf rule, okay, that if adenine is there, there must be thymine. Okay. Now let me assume that this is a helical structure of DNA. Okay. Just say that from here to here. Okay. Just say one helical turn. One helical turn. Okay. So in one helical turn, there will be 10 nitrogenous bases. I would say that 10 base pair. Okay, there will be 10 base pair. So just say this is the pitch of the helix. This pitch is 3.4 nano. This is 3.4 nanometer. So in one helical turn, there will be 10 base pair, and the pitch of that helix will be 3.4 nanometer. So just say that from here to here, there is adenine, and this is thymine, this is guanine, and this is cytosine. Now between these two, the distance between these two consecutive base pair, okay, will be 0.34 nanometer, and the diameter is, this one is 2 nanometer. Okay. Now this is the third point, that let me write here, one helical turn is equal to 10 base pair, it is 3.4 nanometer, and the distance between the two consecutive base pair is distance between these two. Okay, it can be between these two also. Just say TA. So between these two also, its distance will be 0.34 nanometer. So the Watson and Crick, through this model of DNA, many new ideas. Okay, the studies of the genetic information. Okay, through this model. Now we know a lot of discoveries. Okay, medicines have been developed. Okay, so this theory is very important. This postulate is very important. And after these postulates, the Watson and Crick had explained. Francis Crick have come up with a new idea. Okay, with a new idea of that idea is known as central dogma of molecular biology. Francis Crick had given this process. Okay, once after this structure of helical structure of DNA explained by them, they got a Nobel Prize. Okay, sharing with even with Franklin. Okay, it was Watson, Crick, and Franklin. They shared the Nobel Prize on the structure of the DNA. Now next we are going to discuss about the central dogma of molecular biology. DNA to DNA. Okay, this has to be replication. Replication. DNA to RNA we call as transcription. RNA to protein, we call as translation. So this is said to be the central dogma of molecular biology. Okay. So every living cells, okay, first they have to go for copying of DNA, that is DNA to DNA. Then they have to convert to DNA to RNA and RNA to protein. Okay. So if a virus, for example, infect in a human cell, it means that its genetic material will multiply. So if its genetic material multiplies, it means that our body, which has a genetic material, unable to produce useful protein, okay, because the virus genetic material started multiplying, and they will start producing their own protein, and which in other way it affects the immune system of our human being. So normally an HIV, a person get HIV because of this reason. It means that if the central dogma of molecular biology is affected, it is going to affect the other essential processes that is occurring, okay, inside our living cells. Okay. Now with the idea of this central dogma of molecular biology, we are going to understand, we are going to see the process called as replication. What is replication? Replication is nothing but copying of DNA to DNA. To understand this process, we have to understand some of the basic requirement, okay, to undergo replication process. The first is enzymes. The first enzyme is phosphorylase. Phosphorylase. Second is said to be helicase. The third is single-stranded binding protein. Next is topoisomerase. Fifth one is ligase, and the most important one, 6 primase, 7 DNA polymerase, 3, 8 we have DNA polymerase 1. So these are essential enzyme that is required in the process of replication. In the next class, we'll discuss about the steps that is involved in the process of replication.